Thanks everyone for logging on. Just to introduce myself, my name is Erin Judge. I'm the Program Manager for Alumni Events at the College of Engineering and Applied Science. I'm very excited to be here today with Associate Professor Wil Srubar for this alumni webinar. As part of the alumni webinar series, we aim to showcase some of the amazing research that is happening at the college and bring our faculty directly to you so that you can get an exclusive look into their work. Now it's my pleasure to introduce Dr. Wil Srubar. Dr. Srubar is an Associate Professor of Architectural Engineering and Material Science at the University of Colorado Boulder, where he leads the Living Materials Laboratory. He holds a PhD from Stanford University, as well as a Bachelor of Science and Master of Science degrees from Texas A&M University and the University of Texas at Austin, respectively. Dr. Srubar's academic research integrates synthetic biology, polymer science, and cement chemistry to create biomimetic and living material technologies for the built environment. He's received more than $6 million in sponsored research funding through the U.S. National Science Foundation, ARPA-E, and DARPA's Biological Technologies Office. He has authored more than 70 technical journal articles, book chapters, and conference proceedings. His work has been highlighted in the New York Times, The Conversation, and the Huffington Post. He is a co-founder of Minus Materials and Arimus Earth, and he is actively involved in the American Concrete Institute and the American Ceramic Society's Cement Division and their Architectural Engineering Institute. Professor Srubar, thank you so much for taking the time to share your amazing work with our alumni and friends today. I'll go ahead and turn it over to you.
Thanks, Erin. Really great to be here today. Thanks everyone for tuning in. I see some very familiar names. So Torin, Melanie, Lauren, Jesse, and I'm sure there are a few others. It's good to see you. Hope y'all are all doing well. So the title of my talk today is a little provocative toward genetically programmable architecture, and I'm hoping to convince you by the end of this talk that we're not too far away from genetically encoding into the DNA of organisms an ability to grow our building materials that are very responsive and reactive to different stimuli within the built environment. But before I launch into some of our projects, I think it's really helpful to talk a little bit about how I view the world. So, today I'm an associate professor here at CU, but that was not always the case. So I grew up on a cotton farm and cattle ranch on the plains of Southeast Texas, where really the land was our livelihood. Many people don't believe me when I tell them I was a farm kid, but here is living proof that I was. And growing up on a farm every single day, I came face to face with what I would call, the clever ingenuity of nature. And I was always fascinated by nature's response, especially in times of very acute crisis. So I remember, during flash floods where I would walk outside and see colonies of ants just have this innate instinct to pile up on top of each other in order to ensure the colony's survival. I remember, chasing lizards and make them shed their tail, so their little wriggling tail would be left behind to evade, me, the predator. So I was always, very fascinated with nature and how it has evolved these instincts and responses, especially, like I said, in times of these acute crises. So I had a very rural upbringing, but it was the skyline of Houston that really inspired me to become a structural engineer. And so I remember going to baseball games with my dad at the Houston Astrodome and just marveling at how women and men can build such magnificent structures. So I was hooked, with my first baseball game. And this really kind of came full circle. One of the first projects I worked on as a practicing structural engineer with Walter P. Moore, which is a structural engineering firm in Texas, was the Dallas Cowboys Stadium. So it was a really nice return to my roots and my inspiration for me choosing this as a career. You know, when I got a bit older, I moved into concrete jungles. So this was the first time that I really came face to face with other people that didn't have rural upbringings. They lived in cities their whole life, and it was really a light bulb moment for me that I was the generation of my family that urbanized, that moved into cities. And I just remember seeing, you know, we have this image here of Central Park in New York City too. I was always, it came as quite a shock to me that what was the built environment was very separate from what was the natural world. And this is a very clear example. You know, humans were just so separate from nature, which was very, you know, quite foreign to me. So, you know, since moving to these cities and beginning my professional career, you know, I've always really questioned that convention. Why are humans so separate from nature? Why do living things grow, regenerate, and adapt, but buildings do not? You know, they're quite static and sterile. And you know, what can we learn from our fellow species? So at CU, I founded the Living Materials Laboratory, and my group engineers biomimetic and living materials for the built environment. So we integrate aspects of synthetic biology with traditional polymer and cement chemistry to create responsive, adaptive, living materials specifically for the built environment, so for buildings and for infrastructure. In order to do this, we work with a variety of very talented collaborators at CU and even beyond CU. And you know, everything I'm going to show you in this presentation really is I'm indebted to not only them, but also my students that you see here. So my students come from a variety of different backgrounds, from architecture, physics, chemistry, civil engineering, microbiology. And of course, this brings not only opportunities for inventing new materials, but also a variety of different challenges with communication, with perspectives, and really being comfortable with failure. It's something that, you know, I really owe it to my students for coming on this journey with me. And you know, we confront failure every single day in the lab, and occasionally we're quite successful, which is really exciting. So we work on a variety of different material systems, and what I want you to take away from this slide is that, you know, with everything that we do, we really try to blur the boundaries between the built and the natural worlds. And we are really trying to bring building materials to life. We work on a variety of foams and alternative cements, biological adhesives, algae, lichen, and proteins. And really what we're trying to do is fuse natural and biological principles into everything that we do. So Aaron mentioned our sponsors. I also want to thank our sponsors for going on this crazy journey with us as well. You know, I don't have a lot of time to go into every single one of these projects, but I want to highlight 3 of them over the course of the next half hour or so. And the first one I want to highlight is our work really trying to engineer. It's a good showcase for us trying to engineer a biomimetic response in an otherwise non-living material. And so this is our project working with antifreeze proteins. So the motivation for this work is quite simple. We know that ice is very destructive to concrete. Concrete's the second most consumed material on Earth after water. We use a lot of it. We use a lot of it in buildings, and we use a lot of it in roads. And so all of you civil engineers on the call, you know that it's very the process of ice freezing within porous materials like concrete can be very destructive. So we see this evidence of concrete that has you know, spalled off. The surface has basically been torn off because of that action of ice freezing. So water can infiltrate into the material, and then when it freezes, ice expands, creates pressures, and that pops off the surface of the concrete. Now, current prevention methods, because there are prevention methods, but my lab views these as very non-optimal. So for those of you who don't know, for the last better part of a decade, we have put in tiny little air bubbles in concrete to alleviate the pressure that builds up when water starts to freeze and ice expands. These are called air entraining agents. And this technology, like I said, hasn't really changed in the last 70 years. So if you were to look at concrete under a microscope, you'll be able to see these tiny little air bubbles that have been entrained throughout the microstructure, again, to alleviate that pressure. So these air entraining agents are certainly they're really good at alleviating this pressure, especially if you have more than 5% of air entrained in your concrete. It does really well. And so for freeze-thaw climates like Boulder, like Colorado, where we see a lot of freezing and thawing, this works quite well, but it has some downsides. Number one is that air cannot carry load, so it inherently weakens the concrete. Number two, which is can be of concern for departments of transportation, is that when you put in the admixtures to make the air bubbles, it can increase the time of set of the concrete and delay construction. And the biggest one, number three here, is that it increases the permeability of concrete. So in places where you're also experiencing freezing and thawing, you're probably also worried about ice forming on the roads, so you're salting the roads. And these tiny little air bubbles act as super highways for those salts to come into the concrete and cause other damage, like chloride-induced corrosion within the concrete. So good solution, but it has some consequences. We took a step back and said, okay, how does nature deal with this problem? We took inspiration from what are called ice-binding proteins. Now, ice-binding proteins are a class of proteins that are naturally occurring. They are found in a multitude of bacteria, fish, plants that thrive in sub-zero temperatures. And so what these organisms do is that they produce these proteins, and when an ice crystal nucleates inside of them, the protein binds to their surface to keep the ice crystal really small. So this is one of the mechanisms by which these proteins act. This is called ice recrystallization inhibition. Fancy word that just means we keep the ice crystals small and not very destructive to the organism. So what you're looking at here are ice crystal grains without the protein, but with the protein, the ice crystal grains are kept really small. A lot of minerals and ice likes to grow from small crystals into larger crystals to coalesce. It's thermodynamically favored, and the ice binding proteins just simply prevent that by binding to the surface. The second mechanism what we can observe is that when these ice binding proteins bind to the surface, they can change the shape of the ice crystal. So we get these really cool bipyramidal ice, or in the most cases that we're looking and most proteins that we're looking at, we get this like lemon-shaped ice, which is indicative to us that it is in fact interacting with that ice surface. So this is great. Why don't we take some protein and put it into concrete and see if it keeps the ice crystal small and non-destructive. If you know anything about biology, proteins can get very unhappy when you take them out of their native environment. So a normal protein looks like this nice coiled, it has primary and secondary structure, beta folds and beta sheets, and it can have some helices. But when you put it in a non-native environment, it can unravel and denature and even disintegrate. And some of those denaturation agents are things like pH. If you increase the pH, or if you increase temperature, if you increase ionic strength, proteins get very unhappy. If you know anything about concrete, you know that if you squeeze concrete and a drop of liquid comes out, the pH of that solution is about 12.5 to 13. It's very, very basic, high ionic strength. And so, we thought, well, proteins probably won't be very happy in that environment, but we tested it anyway. So what we did is we looked at that solution. So we took simulated pore solution of concrete, and we tested whether or not a protein, an ice-binding protein, has this IRI, this ice recrystallization inhibition activity. So this is the data that you would get. So this is for samples or for solutions, a pH 8.9 solution without any protein. So what we do is we take a drop of solution from a prescribed height, we flash freeze it to get a very thin monolayer of ice. We put it on a cold stage and we watch the ice crystals. And we can see the ice crystals at the very beginning, they're very small, and then over time grow into larger crystals. And so this is a sample, like I said, without any protein. So we see what we would expect to see, that larger crystals grow at the expense of smaller ones. And so this is just blank solutions without any protein. And so we saw what we would expect to see as we increase the pH to higher and higher and higher pHs, and we saw this ice recrystallization inhibition. When Elizabeth was my my main PhD student on this project, when she loaded the sample with an ice-binding protein from an Antarctic bacterium, what we saw was pretty exciting, was that after 30 minutes, the ice crystals remained small, indicating to us that this protein maintained activity at pH roughly 9. We were exceptionally excited to see it maintained that activity at pH 12, pH 12.7, but then after we reached a certain threshold, what we proved in these papers is that the protein unraveled and then it disintegrated because the pH was so high. We also tested another ice-binding protein that we thought would be a little bit more effective in this calcium-rich, high pH solution, but we found the same thing. So, even though it worked up to 12.5 pH, pH of 12.5, it's still expensive. So we knew that we couldn't, this couldn't be a viable option to put into concrete. So we turned to biomimicry instead. So we looked at polymeric molecules that display the same ice recrystallization inhibition activity as the proteins. And in the literature, we found that polyvinyl alcohol was a polymer that displayed this activity. The issue with polyvinyl alcohol was that it had really low solubility in water at high pH, which would not work out well for us. And it has what is called a high cryogelation potential, meaning that if you freeze and thaw it, it forms a hydrogel and wouldn't behave the same way as it would be just dissolved in solution. We took note of PEG, polyethylene glycol, that had the benefits that PVA did not. The only downside with PEG was that it was not IRI active. So we thought if we combined the two into a copolymer, we would either get the best of both worlds or the worst of both worlds. And fortunately, because I'm highlighting it, we did get the best of both worlds. So we did prove, and I'll show you some data, that PEG PVA is IRI active at high pH. We can dissolve it in water, and it gets us the performance that we wanted. So PEG PVA displayed IRI activity, not showing the dynamic ice shaping, but it did that as well in these really high calcium-rich pH solutions, pH 13 solutions. So over here on the left, we have a control sample after 30 minutes of incubation on the cold stage with no polymer dissolved in solution. And you can see the ice crystal grains are actually quite big. PEG alone at a low concentration and a high concentration didn't display this ice recrystallization inhibition, but PVA at low and high and PEG PVA at low and high concentrations did. And it kept the crystal size more than 50% smaller than the blank, the control solution. So we were really excited about this. and thought, okay, well, let's go ahead and put this molecule in cement paste and see if it resists freezing and thawing. So this is results. This is microcomputed X-ray tomography images, basically an X-ray cross-section of cement paste samples that underwent freezing and thawing for 30 cycles without. So on the left-hand side is the sample without the molecule. And then again, these samples here are with the molecules at low and high concentrations. And what we were really excited to find was that PEG PVA, when added at a slightly higher concentration, did exhibit enhanced freezing and thawing compared to the control samples and all of the other samples that had the molecules in them. So this was, this indicated to us that we should go ahead and scale this up to full-scale concrete, which is what we did. So does biomimetic PEG PVA prevent freeze-thaw damage in actual concrete? This is what Mo, my PhD student, and Shane also recently, found the answer to this question. So what you're looking at here is data from a freeze and thaw test of concrete. And on the x-axis is number of freezing and thawing cycles. So we made samples of concrete, exposed them to freezing and thawing cycles up to 300. And what we were measuring is the relative dynamic modulus of elasticity. So what we were actually measuring is kind of the integrity of the sample through the thickness. And we were, again, comparing that to a pristine sample that was not, didn't have any exposure to freezing and thawing. And what we found was that the control sample without any air entrainment or without any molecules quickly failed after about 100 cycles, which is what we would expect. We went ahead and put air into one of the samples. So we compared this to an air entrained sample. And we also found what we would expect to find was that the air entrained sample also resisted freezing and thawing. Nothing new here. But the really exciting finding was that our PEG PVA sample also exhibited enhanced freeze-thaw resistance, looked identical to the air entrained sample, but I'll draw your attention to this 2.6% here. Our molecule, when we use it, does not entrain air. So what we proved in this study was that our molecule was behaving very differently from an air entrainer. It wasn't because it was releasing the pressure that built up from the ice formation and expansion. It was preventing the ice crystals from growing. So this really represented a complete departure from 70 years of air entrainment technology by offering a different route to enhancing the freezing and thawing resistance of concrete. We're really exceptionally excited about not only the applications of this technology to concrete. Oh, by the way, these are kind of the visual images of the samples where you can kind of see our PEG PVA concrete really looks identical to the air entrainer. I'm going to skip this really quickly. But we're really excited about applications of this technology to other industries. So food security, where we're having to store our food, warfighter performance in very cold climates, energy applications, creating anti-icing surfaces for enhanced efficiency of wind turbines, and even medicine. So when we cryo preserve organs and tissues, et cetera, freezing is certainly an issue in all of these industries. The next project I want to highlight is our work with programming biology to make materials for us with very tunable properties. Now, the impetus of this work came about as my group was reviewing self-healing concrete. Many of you may kind of come across self-healing concrete in the literature, but microorganisms have been put into concrete to impart a self-healing characteristic. So the idea being that these microorganisms that undergo biomineralization, they make minerals, primarily calcium carbonate, that, when a crack forms, if these organisms are inside of the concrete, that they would generate calcium carbonate and they would seal the cracks. There's an issue with this because concrete's a very aggressive environment, like I already said beforehand, and the microbes don't always survive at levels that we would need them to survive if we want them to be viable for decades. But again, this concept of using microbes that biomineralize has been applied to the self-healing concrete field. It's also been applied to stabilize soils, and it's also been applied to densify recycled aggregates. So if we bust up old concrete and we want to use it as the rocks in new concrete, some researchers had looked at using microbes to help biomineralize and densify that aggregate to improve the aggregate quality. Again, taking a step back, all of these applications leverage this process of microbial biomineralization. And we have, up to this point, relied on wild type bacteria that do this normally out in nature, to perform in this activity in these applications. But we know that biomineralization is a metabolic process that is governed by the biosynthesis pathways that are encoded within the genome of that organism. So there's this grand opportunity for us that if we understood the genome sequences that enabled better mineral properties, we could really use that to our advantage for these and other applications. So yes, I'm talking about genetically modified organisms, but not for food production, but for engineering them to produce materials for us. So to do that, we have to sequence a genome and understand the genome. And fortunately, the cost and time of sequencing a genome has plummeted over the last few years. And I'm just comparing this to Moore's law. Many of you are familiar with Moore's law, that process speeds will double every 18 months. And just comparing that to the cost of sequencing a genome, it doesn't even follow Moore's law. It was much faster at reducing the cost over time. So what this tells you is that, for a couple $100, we can have the genome of virtually any organism on Earth. And so it's now feasible to decode the genome. And now that enables us to design fragments of DNA to infuse those into organisms to make better materials for us. So since this discovery and since the plummeting of the cost of sequencing a genome, synthetic biology toolkits have really enabled biofabrication of a variety of bioproducts. The definition of synthetic biology as a field is a field of science that involves redesigning organisms for useful purposes by engineering them to have new or enhanced abilities. Up until this point, organisms have been used to make fuels or chemicals, even pharmaceuticals. So most insulin is made by hijacking the metabolic process of microorganisms to get them to produce insulin that we use with people with for people with diabetes to use. And only now are we really scratching the surface with how can we harness these processes to make better materials for us. A lot of companies have sprung up trying to use synthetic biology and microorganisms to make a variety of different products. I'm highlighting Bolt Threads and Ecovative, who are looking at making building materials, so applications to the built environment. So my highly interdisciplinary collaborators and I asked a question. Could synthetic biology help us program bacteria to architect different biominerals with different physical and mechanical properties? So can we really biologically architect minerals by controlling the DNA? And what we're getting at is moving away from the classic material science triangle of processing, structure, and properties, really infusing in genomics and DNA manipulation into that entire process. So we conducted an experiment with a native biomineralizing organism, S. pasteuri, which has been used a lot in self-healing concrete literature. And then we engineered 2 strains. So working with my very talented collaborators, we took E. coli and we integrated either a single copy or multiple copies of the urease gene cluster, which ultimately governs biomineralization in these in ureolytic organisms. So we imparted that ability to E. coli, and we wanted to look at the differences in the DNA. So, you know, is putting in multiple copies of that gene cluster better, or is a single copy better, and how does that compare to a regular native producer? So we grew these in their respective media conditions. They made minerals, and then we looked at the minerals and we analyzed them. And what we found, it was just this crazy morphological diversity between the native and engineered strains in terms of their ability to architect different minerals. So from very rhomboidal to very globular, and the sizes of the crystals also varied. You know, so what we ultimately concluded was that genetics, you know, nature certainly plays a role in the size and shape and even the mechanical properties of these minerals. So what you're seeing here is nanoindentation, basically a measure of the stiffness of the crystal. Nanoindentation results comparing the minerals of the native producer here in green. And then what you're seeing in red is the single copy, the E. coli with the single copy of the gene cluster, the mechanical properties of those minerals that organism made versus the mechanical properties of the minerals that the E. coli with multiple copies, you know, what that exhibited. And what we really found was that slow and steady wins the race. If we actually just imparted a little bit of the urease activity to these organisms and put them in the right environment, they would make stiffer crystals. And it's clear, you know, the red and blue really highlight the differences that the DNA imparted to the ultimate mechanical properties of the organisms. You know, this finding was really intriguing to us since it speaks to the potential of genetically encoding the architecture of building materials and even possibly buildings into the DNA of organisms. You know, of course, we have a long way to go, but you know, nature creates these very complex, hierarchically ordered materials. And just think of shells, you know, seashells and coral. And for the first time, we are now able to harness that potential and control the hierarchical growth of those materials by manipulating and controlling the DNA. Again, it might seem like science fiction, but the reality is a lot closer than one might think. So the final project I want to highlight is very related, and it's our work in which we are bringing building materials to life. So my lab and my collaborators started to rethink self-healing concrete. You know, like I said, self-healing concrete is a concept in which we take regular concrete and we put in this biomineralizing organism into that cementitious matrix. But prolonged cell viability in that very harsh environment is not conducive, or that harsh environment is not conducive to prolonged cell viability. And in fact, no researcher has been able to prove that more than 10% of the initial inoculum of the bacteria survives more than 7 days. So it doesn't seem like concrete, which we expect to last decades, if not a century, in you know, today, that this is a viable path forward. So we asked, could we leverage these biomineralizing organisms to create an engineered living cementitious composite by giving it the right environment by which to thrive, to make minerals, and even survive. So our goal was to grow a living structural material using this biotechnology and to keep it alive, have the initial inoculum, you know, more than 70% of it survive at 7 days and longer. And this is what we came up with. So we took biomineralizing bacteria and we encapsulated it within a sand and hydrogel scaffold. And how we did this was we took sand, hydrogel, the media that the organisms need and the cells, and we mixed it all together. And inside, as these organisms were growing, they were precipitating these biominerals that were helping to cement the sand particles together. There were also this hydrogel that was kind of helping to keep all of the moisture inside of the material, especially once we cooled the material from an elevated temperature. Cells generally like to grow a lot faster the higher the temperature. But once we cooled the material, we formed kind of a jello around the cells that had all of the minerals inside of it and the sand. And by the way, this is published in the journal Matter, if you want to read a little bit more technical details about it. But this is what it looked like. So this is post-gelation, right when we popped it out of the mold. And this material is just teeming with living bacteria that biomineralized inside, in between the sand particles. And then we were able to control the desiccation of that material and impart better structural properties with decreasing humidity. So there was this trade-off with keeping it gooey and squishy and keeping the bacteria alive versus drying it completely out and getting really good structural performance. We're able to make very large samples. By the way, this work was sponsored by DARPA, so that's why there's some propaganda with this brick. But we are able to make very large samples, and we can make virtually any shape and any size. And I'll show you some more examples of this. But if you, again, take a half a step back and now view this material as a platform, what's exciting is that different hydrogels and different bacteria that we can encapsulate could impart multitude of different functions to otherwise inert building materials. So, one thing that we've been playing around with was encoding secret messages, because certain bacteria will fluoresce under different types of light. And so if you pattern them appropriately, you can start to display, secret messages that can be interpreted under different light conditions. Or you could impart, bacteria that sense and respond in other ways, maybe sense chemicals or toxins in the air and then impart something back to neutralize it. We've looked at photosynthetic organisms that could impart enhanced CO2 sequestration in service. So, while we have this initial, what we would consider a platform material, we just see this as a tremendous starting point for engineering other types of living building materials with a multitude of different biological functions. This is for my civil engineers in the audience. The number I'll point out is the strength of the living building material, which is about 4 megapascals. If you're comparing the compressive strength, it's on par with a cement-based mortar, so the mortar that kind of surrounds the bricks in everybody's house. So not ultra high strength concretes, but also not trivial in terms of the compressive strength we're able to achieve. I'm going to fast forward here. And so the last, I think one of the last things I'll discuss is that we actually regenerated the living building material and demonstrated in our publication that one parent brick we can keep it alive and it can grow up to 8 child bricks without the addition of new bacteria. So what we did is we made one living building material, what we call the parent generation. We split that into 2. So we took 2 halves and we added more abiotics. So not new cells, but we added more sand and we added some more media. And over a short period of time, the bacteria grew 2 full bricks in each of them. Again, bacteria grow at an exponential rate. It didn't take very long, a couple of hours to a day or two to have a fully formed first generation of 2 bricks from one parent generation. And we did that 2 more times. So we grew 8 full child generation bricks out of that one parent generation. And what we just wanted to demonstrate was a potential for this on-demand exponential manufacturing, especially in resource-limited environments. I saw that someone submitted a question, beforehand for me to answer about Mars, and this is a great opportunity to bring this up, because we really do think that this technology, sure, there's some applications on Earth in resource-limited environments, like out in the desert or in the Arctic, Antarctic, where there aren't a lot of raw material resources to make materials. But we also have our eye on the moon and on Mars, where we do have sources of sunlight and we have sources of CO2 even on Mars. So if you use photosynthetic organisms to grow materials for you, this is a great technological solution to make the materials and to make them at a very fast and exponential rate, which is pretty exciting. This is just to show you that through the parent and child generations, the bacteria stayed alive if we kept the humidity above a certain threshold. So here is just a measure. Most probable number is a measure of how many bacteria are alive inside of the material. So obviously, if we kept the humidity relatively high at 100%, all of the bacteria survive through the parent, first, second, and third child generation. The same was true for 50% relative humidity, which is even lower than what the humidity is in most buildings. We keep the humidity at about 55% and up to 65, maybe even 70%, kind of narrow window where humans like to be comfortable. And obviously, if we dried it completely out after the second generation, the bacteria ultimately died because they were super stressed from a lack of access to moisture. And all the while, as we kept growing them through the generations, they were mineralizing more and more and more and creating more mineral. So this is just to show you that the quantity of the mineral on the left-hand side increases as you go across through the generations, which is super exciting. The bacteria were not only alive, but they were functional in creating this biocement, helping us create the biocement for us. So yeah, we're able to make a variety of different shapes and sizes. That's not necessarily the limitation. We've experimented a lot with 3D printing and have plans to do more demonstration projects related to that. A civil engineer did not necessarily make this shape because it's not something that we would make, but it's to demonstrate complex shapes that are achievable with the material. And this toys with this idea of going from living materials to living structures, because any structural engineer will tell you that efficiency really comes not only from the material you use, but the shape in which it takes. So if we can better understand the loads imparted on the structure and the material properties, we can design the most efficient shape for that application. My dog is very excited about topology optimization, so apologize. But we've gotten a lot of press lately, both me and my collaborators. We're really indebted to the New York Times and to even Science Friday and NPR for really helping us spread the word on our technologies. So this is just a glimpse in my view of the world. I really do think it's an exciting time to be an engineer. I think it's an exciting time to be a civil engineer, especially one that has an interest in interdisciplinary work. Because I think, in general, we as engineers are facing unprecedented challenges, looking at increasing strength and frequency of disasters to social inequities and to climate change. So I really do think that, if we look to nature, we're not the only species that's having to adapt to climate change. And I really do think that if we pay more attention, we do have a lot to learn from each other. So with that, I wanted to thank Aaron and Kevin for inviting me to share a little bit about our work, and happy to answer any questions that any of you may have.
Awesome. Thank you so much, Wil, for that presentation. That was great. in the chat, and for any of our participants, please feel free to use that Q&A button at the bottom to send in questions for Wil. So we'll start with this one. It's from Tristan. He asks, why wait? How can this be integrated into current tenant improvement or ground up construction projects in Silicon Valley?
Yeah, we're trying. We have some great demonstration projects here in the lab and pilot scale demonstrations that are being funded by the Department of Defense, which is really exciting. I think the challenge that we face and we're spinning this out into this living building material. We're spinning this out into a company called Prometheus Materials. So, we're some years away, but and there are some regulatory hurdles that we have to get past with introducing organisms into an industry that there's an inherent negative connotation with microbiology and the microbiome in your homes, and we want to change that perception. There are some trailblazers that we're following. So companies like Ecovative, which has a fungal insulation material grown from mycelium fungus, Biomason, that's creating a tile brick out of a similar biomineralization type process. So, we're certainly on the way, but it'll take a few years for us to get to Silicon Valley and actually build a building. Yeah.
Great. Another question, this one is from Roger. How can end of life of these new materials be safely addressed? Can they be recycled without harm to the environment?
Yeah, absolutely. What's really important to us is that we use environmentally benign organisms and environmentally benign molecules. I'll start with the molecules. So the polymer PEG PVA that we use inside of concrete that displays that ice recrystallization inhibition, that's actually a molecule used in drug delivery. So it's biocompatible, and it's actually it's very robust. It's usually used to increase the circulation time of drugs in the system. So it's not easily broken down, but it is biocompatible and bioresorbable over time. So if that molecule were to ever leach out or to get into lakes, rivers, streams, we're actually not even coming close to the environmental consequences of actually salting the roads. That's actually very environmentally destructive. And we use it in such low concentrations that it would never become a problem. Now, the living building materials, and we get this question all of the time with using organisms, and we also try to stick with organisms that are not heavily engineered, that are normally found in nature, and we just boost because there's kind of two strategies of tailoring and tuning mechanical properties using biology. One is to do what I showed here, which is go in and manipulate the DNA to make a brand new mutant that has never done this before. The other is to apply selective pressures to organisms in kind of it's a very Charles Darwinian natural selection process where you apply different pressures and select the natural mutants that occur over time for the best performing, and then you grow up that strain and you use it. And we prefer that. We certainly prefer that because that's going to lower all of the regulatory issues that we have with applying genetically modified organisms to the built environment. And so in terms of recycling, that's important to us as well. The more benign the material is and the more easily we can break it down, we're trying to question that paradigm too. I show you how we split the brick and regrew it. You can think of that as a recycling scheme as well. We're trying to really question the paradigm of why make a material and have it last 200 years? Why can't you just have materials that kind of regenerate on the normal cell life cycle? I think that's not a horrible thing, but it's going to take a lot of convincing.
Thank you. One more question here from Dan. Can you speak more about the genetic control of biological branching systems?
Yeah, Dan, this is really great, a really great question. And a lot of my, I would say, biomedical and chem bio colleagues know a lot more about this than I do, because for us, we're really interested not necessarily in the cell, but in the materials that it makes, and we're really focused on bacteria. But there are mammalian cells, right, used in stem cell research, and there are plant cells. Again, two categories of cells that we haven't yet explored that do differentiate, and you can control that differentiation through the number of cell divisions. So there's like a disappearing sort of trigger that once a cell divides a certain number of times and that signal disappears, that it all of a sudden becomes, starts producing or starts differentiating into a different type of cell. And that happens in mammalian cells and it happens in plant cells. There's some great plant science research too that shows that even mechanical stressors can get a cell to differentiate and to branch into different types of cells. But we haven't yet scratched the surface on that. I'm looking to our colleagues to blaze the trail on that differentiation, because that also will enable us to move away from just living materials to living structures if we can get these living cells to architect different shapes and patterns all on their own. So it's an exciting field, and we have a lot to learn, especially in this very interdisciplinary space.
Thank you. Next question here from Jesse. How do you think lighting can best serve these living buildings alongside lighting service to the living occupants?
Yeah, good question, Jesse. And lighting is something that we've looked, well, I have a lot to say on this, but we've looked at other materials that will help with lighting. We looked at transparent wood. That's certainly not living, but could enable diffuse daylighting and help with those types of applications, getting away from high energy silica glass and other, highly insulated windows. But in terms of the living building materials, we haven't really looked so much at light triggers or the need for light. We've toyed with the idea of light transparency in the living materials. So there are certain, bacteria certainly that form biofilms that are a little bit more translucent, and we're excited about that space. There's a really cool artist making fungal, it's kind of like diffuse indoor light coverings that actually are grown using fungus. So what you get is this beautifully patterned, greens and oranges and browns from the living fungus within the planes of glass. So this like intersection of lighting and living building materials, it's just now starting. So I think these types of conversations and questions just get those creative juices just flowing. So Jesse, if I can convince you to come back and get a PhD, maybe we can work on that together.
Great. And final question here before we close it out. This is from Sebastian, asking, how are the living bricks that regenerate the child bricks able to regenerate complex shapes? Do you have to separate out the bricks after it reaches a certain size?
Yeah, I think, oh, hey, Sebastian, I know Sebastian too. So I think if I understand your question, it's, how do we get this, half of a material to form like a different shape? And maybe I should explain the process of how we regenerate. So, we cut the material in half. So we split the brick, and then we do put it back into a mold, and then we add the other abiotics. So that mold, you could crumble up that half of the material and spread it out throughout the mold, and add the abiotics, and it could be formed. It could certainly form that way. And that mold and the sand really creates the dimensional stability by which we can grow virtually any shape. The material doesn't grow out of control. We always get this question too, like, will my house turn into a skyscraper? And will your materials just help increase the value of my house by increasing the square footage? But the answer is no. So the bacteria are encapsulated within within the matrix, so it doesn't grow out of control. So we don't have to separate out the bricks after it reaches a certain size because it's that stability of that mold that keeps it from growing out of control. I think I answered. I think that was the answer to that question.
Awesome. Well, I want to thank you again, Wil, so much for your time today. This was really exciting research to go over. And just before we log off here, everyone, you'll notice on your screens, we have launched a poll asking you to just indicate on a scale of 1 to 10 how likely you are to recommend another CU Engineering alumni webinar. We really appreciate your responses on this. And additionally, please be on the lookout for a follow-up email tomorrow. We'll send out some additional information on how you can stay connected to the college, as well as some of our upcoming engineering alumni events that we have. We'll also include a little deeper survey there so that you can share some additional feedback on this webinar with us. So thank you again, Professor Srubar. Thank you everyone for participating today. We really appreciate your time and hope to see you all again soon.